The homologous chromosomes, chromosome 1 from mom and chromosome 1 from dad, both duplicate. So now you have sister chromatids, you have 4 chromosomes. Two homologous chromosomes here, sister chromatids here, also homologous chromosomes. Make sure you keep those clear. I know I don't say it very straight. Make sure you get that clear in your mind. So this is interphase before meiosis. So now during mitosis, the duplicated chromosomes separate from the sister chromatids separate. So each daughter cell gets the correct chromosomes. In meiosis, it's a little bit different. The homologous chromosomes actually separate. So you get mom's genes going one way, dad's genes going another way in the cell. So each of these cells has 46 chromosomes, but it's not diploid because it doesn't have, it only has 2 copies of the sister chromatid, or has a sister chromatid. It's not homologous chromosomes. And those will split in another cell division. Now you'll get gametes with 1 copy of each chromosome. Alright, so during the process of meiosis, a couple things here. Haploid cells are produced through 2 cell divisions. Interphase, well, you have an interphase followed by those 2 cell divisions, meiosis 1 and meiosis 2, and something called crossing over occurs. We'll talk about that in just a second. We'll watch our handy-dandy video and then we can talk about it.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells. Cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
Alright, so I don't know if the honor code is broken there, but I don't enjoy heading into that guy's pants like that. So I apologize for that. But, so a lot lot of stuff there. I hope you noticed a lot of similarities to mitosis. It is essentially the same processes with a few slight differences. And so, luckily, you catch a break there. You don't have to learn a whole new process as it is similar to mitosis. So, let's go through each of the stages real fast. Interphase, the chromosomes duplicate, similar to mitosis. The homologous, make sure you know the homologous chromosomes versus sister chromatids. You get a homologous chromosome or a sister chromatid from your father and your mother. Which one is it? You get a homologous chromosome from your father and one from your mother. Sister chromatids are the duplicated exact copies. Alright, so the centrosomes will be there, but they won't form the spindle yet. So this is interphase. Prophase, chromosomes coil up. They will, so this is slightly different than mitosis. The homologous chromosomes pair up so that they can have their crossing over event. So this is called genetic recombination. This is why your brother may have, or you may have your mom's chins and your dad's eyes. Chins, your dad's mom's chins. You have one. But this crossing over, it exchanges the same amount of DNA on each chromosome. So it's not uneven, it's always an even switch. So each chromosome will retain its normal size. Nuclear envelope will fragment, and then the spindle will start to form. So nothing new there. Here, the homologous chromosomes line up in metaphase 2. So this is a little bit different than metaphase and mitosis. There, the sister chromatids line up in the middle. Here, homologous chromosomes line up and get pulled in the middle through the tug-of-war like we talked about. In anaphase, the homologous chromosomes will separate, and the sister chromatids will migrate together. So, different than mitosis. Make sure you are aware of that. And then telophase, same thing. The cytokinesis will start to occur through the cleavage furrow, and the two different cells will start to form. So now you have two cells. How many copies of chromosomes, or how many chromosomes does each of these cells have?
46.
How many chromosomes do they have?
46.
How many chromosomes?
46.
They have 46, but they're not diploid. They are, because they have sister chromatids, which are exact copies of each other. So I've never found a good way to really keep track of that, but just know they're not haploid just yet. They still have 46 chromosomes which will separate. Alright, so now we can go through this quickly. The second one, there's really no differences. Prophase, you get the centrosomes, or the spindle forming, and you get the nucleus will fragment or disintegrate again. Metaphase, everything will line up. And now here, in metaphase, I shouldn't be there. So in metaphase 2, the cells will now, or the sister chromatids will be lined up in the middle. That's not a very good picture, so I don't know why I have that there. They will start, the sister chromatids will now separate during anaphase 2, and the spindle will start to elongate, pushing the two cells apart. Telophase 2, we have the sister chromatids separated, pulled into each cell. The nuclear membrane will now reform, and the cells at this point are haploid because there was no DNA replication at this point because there wasn't any interphase. So we only have cells that are now haploid. So that's meiosis. Any comments, questions?
So even though they have, like, they're red and blue, they're only one because it's a copy of a red and blue that was already there?
Right. So, yes.
Okay.
Yeah.
So then what happens to the haploid cell after this?
What do you mean? What happens? Where does it go?
Yeah.
Oh, so good question. Where are haploid cells located? In females, they're in your ovaries. That's where your eggs are. And males, in the testes, where sperm are. So this doesn't happen all over your body, and then you don't have like sperm forming in your brain, and they swim down. And they're actually, it's just in very specific spots in your body. So this doesn't happen everywhere. A good question. Any other questions? All right. So back into his pants.
Humans produce gametes, eggs and sperm.